Hello students, welcome to EPG Pathshala. I am Dr. M.N. Gupta, Emeritus Professor from Department of Biochemical Engineering and Biotechnology from Indian Institute of Technology, Delhi. Today we are going to talk on passive transporters, ionic channels, and a disease called cystic fibrosis from paper Structure and Function of Biomolecules 2. We discussed active transport in detail. In this module, we will describe two important examples of passive but facilitated transport systems, and we will see what essentially is the difference between facilitated transport and the active transport. We follow it up with discussion on ion channels. The medical relevance of membrane transport will be illustrated by pointing out two examples. One is glucose transport, which is under insulin control, and a medical condition called cystic fibrosis. So the objective of this module are to learn about various transporters of glucose with emphasis on glucose transporter of erythrocyte as an example of facilitated transport. We will also learn about the role of the bicarbonate anion and chloride anion exchanger protein in the transport of CO2 from the blood to the lungs. To learn about ionic channels and their importance in membrane transports, and finally to understand how a defect in chloride ion channel can cause cystic fibrosis. So the concept map is about the two types of membrane transport in this module: facilitated transport and ion channels. Facilitated transport illustrated by the GluT1 transporter and bicarbonate chloride anion exchanger, and the ion channel illustrated by cystic fibrosis. Membrane transport encompasses diverse biological phenomena, and it is not possible to discuss all transport systems. When we discussed ATPases, we referred to how reverse action is possible for ATP formation. At the same time, many transports are driven by other energy forms like PEP, which is another high energy compound, sodium motive force, and even light. Transport is also involved in nerve function and many signal transduction mechanisms. In this module, the emphasis is to illustrate some important examples about the relevance of membrane transports to diverse basic biological processes and some diseases. The glucose transporter of erythrocytes is an example of passive transport. Erythrocyte metabolism requires that they continuously get glucose supply from blood plasma, which is nearly constant at around 4.5 to 5 millimolar concentrations. The transporter facilitates glucose entry into erythrocytes by enhancing its rate 50,000 times over just diffusion controlled rate. This transporter is different from glucose transporters in other tissues and is called GluT1. It's an integral membrane protein of 45 kilodalton molecular weight with 12 membrane spanning helices. It is these helices which form a channel with hydrophobic inner surface for glucose to pass, forming transient H bonds, hydrogen bonds, during its movement. Glucose transport via GLUT1 follows Michaelis-Menten kinetics, with the rate of entry having a saturation kinetics at high glucose concentration. Given the blood plasma concentration, GLUT1 operates at Vmax most of the times. GLUT1 is specific for glucose with Kt equal to 1.5 millimolar. The specificity is not absolute, but Kt for mannose, D-mannose is 20 millimolar, which is much higher, and 30 millimolar for D-galactose, even higher. In liver, another transporter, GLUT2, facilitates glucose transport out of hepatocytes. Glucose is produced here by glycogenolysis. Muscle and adipose tissues have GLUT4, which is controlled by insulin level. Insulin release exposes GLUT4 on all surface membranes, thereby increasing the glucose uptake by more than 15-fold. The erythrocytes also contains a transporter, which is another one which carries out facilitated transport. This anion exchanger, called the chloride bicarbonate exchanger, enables erythrocytes to pick up carbon dioxide from blood plasma and carry it to the lungs. The CO2 is of course released in the blood from respiration in tissues such as muscle and liver. In erythrocytes, CO2 forms bicarbonate anion, bicarbonic anhydrase. The high turnover of carbonic anhydrase reflects the importance of this step. The bicarbonate anion is now released back by the erythrocytes into the blood. The benefit for this arises from the fact that the bicarbonate anion is more soluble in blood plasma than carbon dioxide gas. In the lungs, bicarbonate anion re-enters into erythrocytes and is converted to carbon dioxide and released again to be expelled by lungs. The efficient shuffling of carbon dioxide across the erythrocyte membranes is achieved by increasing membrane permeability through this transporter. For each bicarbonate ion movement, one chloride anion moves in the opposite direction. Hence, this transport system is an example of anti-porter. GLUT1 was an example of a uniport. If chloride anion transport does not take place, bicarbonate anion transport also does not take place, as the process is a coupled process. This transporter, also called anion exchanger, is also an integral protein which spans membranes 12 times just like GLUT1 transporter. The exchange maintains electrical neutrality and hence membrane potential is not disturbed. NMR studies an inhibition of the exchanger by 1,2-hexane cyclohexane dione. Diene indicates that anion binding causes conformational change, resulting in exposure of the binding site to opposite faces of the membrane. The erythrocytes also contains a transporter, which is another one which carries out facilitated transport. This anion exchanger, called the chloride bicarbonate exchanger, enables erythrocytes to pick up carbon dioxide from blood plasma and carry it to the lungs. The CO2 is of course released in the blood from respiration in tissues such as muscle and liver. In erythrocytes, CO2 forms bicarbonate anion, bicarbonic anhydrase. The high turnover of carbonic anhydrase reflects the importance of this step. The bicarbonate anion is now released back by the erythrocytes into the blood. The benefit for this arises from the fact that the bicarbonate anion is more soluble in blood plasma than carbon dioxide gas. In the lungs, bicarbonate anion re-enters into erythrocytes and is converted to carbon dioxide and released again to be expelled by lungs. The efficient shuffling of carbon dioxide across the erythrocyte membranes is achieved by increasing membrane permeability through this transporter. For each bicarbonate ion movement, one chloride anion moves in the opposite direction. Hence, this transport system is an example of anti-porter. Blue T1 was an example of a uniport. If chloride anion transport does not take place, bicarbonate anion transport also does not take place, as the process is a coupled process. This transporter, also called anion exchanger, is also an integral protein which spans membranes 12 times just like blue T1 transporter. The exchange maintains electrical neutrality and hence membrane potential is not disturbed. NMR studies an inhibition of the exchanger by 1,2-hexane cyclohexane dione. Diene indicates that anion binding causes conformational change, resulting in exposure of the binding site to opposite faces of the membrane. Apart from ion pumps, ion channels guard the entry of movement of ions through cell membranes. There is one basic difference between the two. While pumps have to spend energy to maintain the concentration gradient across the membranes, channels let ions move rapidly as per electrical and concentration gradients. We need these twin mechanisms because of the diversity of the biological processes which involve transport of materials, particularly ion transport. And these diverse processes include signal transduction, pH balance, volume regulation, the cell cycle, fertilization, immune response, secretions, muscle contraction, and electrical signaling in nerves, and muscles and synapses. During flow of ions through channels, transmembrane electrical currents are generated. While with sodium and potassium ions, the result is change in the membrane potential. With calcium ion, the signal is chemical in nature. Ion channels involve passive, rapid and thermodynamically downhill transport. So ion pumps differ from channels in these three respects. A distinctive feature of an ion channel is that it requires only one gate. An ion pump needs at least two gates which cannot be opened simultaneously. So a gate can be visualized as a protein component which controls the ion movement on the basis of conformational change. An ion channel is a hydrophilic path which allows movement of the ion through the hydrophobic environments of the membrane interior. It is selective but energetically favorable transport process. The ion channel exists in open gate and closed gate states. The opening and closing of the gate is dictated by appropriate signals generated by cellular necessities. The ion flow is according to the electrochemical gradient. The rate is so fast that tens of millions of ions are transported per second through a single channel. The opening and closure of the ion channel is also fast and occurs with the frequency of several milliseconds. This reflects the rapid nature of protein conformational changes which operate the gates. A channel often has more than one gate. In such cases, all gates must be open for the transport of the ions to take place. For example, ion channels for sodium ion, potassium ion, and calcium ions have two gates. Both gates must be open for transport of these ions. This is not to be confused with the way gates operate in case of ion pumps. Those gates cannot be opened simultaneously. In case of channel, the multiple gates have to be in open position simultaneously. High resolution X-ray and some functional measurements have confirmed the above picture, which distinguishes pumps and channels. If a pump starts functioning like an ion channel, even for a small fraction of a second, the result will be catastrophic for the cellular functions. The gates thus have a fail-safe mechanism inbuilt in their function. Slide 11. Ionophores are carriers which facilitate ion diffusion. These are organic molecules. Important examples are antibiotics of bacterial origin such as valinomycin and gramicidin A. There are two classes of ionophores. One class is of carrier ionophores, which increase the permeability of ion by binding the ion, transporting it through the membrane and releasing it on the other side. On the other hand, channel forming ionophores form transmembrane pores or channels which allow an ion to pass. Slide 12. Valinomycin is a carrier ionophore. Its transport of potassium ion into the mitochondria uses energy of electron transport chain. Oxidative phosphorylation does not take place and no ATP is formed. The carriers like valinomycin forms a donut type structure in which hydration of potassium ion is prevented by chelation by the oxygen atoms of the antibiotic, as is shown on the right hand side in this slide. The periphery being now hydrophobic, the transport is facilitated. The above function is so selective that binding of sodium ion is 1000 times less than potassium ion by valinomycin. The hydration-free energy of sodium ion is minus 72 kilocalories per mole, and that of potassium ion is 55 kilocalories per mole only. So valinomycin competes less effectively with water for sodium ion. Valinomycin as a carrier ionophore has a good turnover and transports potassium ion many times per second, returning each time to surface to pick up a new potassium ion. High flexibility of valinomycin makes this a facile process. Slide 30. Gramicidine A is a 15 amino acid residue long peptide with alternate L and D amino acids. All amino acids are hydrophobic with both N and C terminals being blocked. X-ray diffraction studies have revealed that the gramicidine A exists as a dimer with head-to-tail joining. The result is a 4-angstrom diameter helical channel for sodium and potassium ion movement. The hydrophobic amino acids allow formation of the nonpolar exterior in contact with membrane structures. Ions enter and diffuse to the other side. In this mechanism, the channel former does not move. Ionophores are carriers which facilitate ion diffusion. These are organic molecules. Important examples are antibiotics of bacterial origin such as valinomycin and gramicidine A. There are two classes of ionophores. One class is of carrier ionophores, which increase the permeability of ion by binding the ion, transporting it through the membrane and releasing it on the other side. On the other hand, channel-forming ionophores form transmembrane pores or channels which allow an ion to pass. Valinomycin is a carrier ionophore. Its transport of potassium ion into the mitochondria uses energy of electron transport chain. Oxidative phosphorylation does not take place and no ATP is formed. The carriers like valinomycin forms a donut type structure in which hydration of potassium ion is prevented by chelation by the oxygen atoms of the antibiotic, as is shown on the right hand side in this slide. The periphery being now hydrophobic, the transport is facilitated. The above function is so selective that binding of sodium ion is 1000 times less than potassium ion by valinomycin. The hydration-free energy of sodium ion is minus 72 kilocalories per mole, and that of potassium ion is 55 kilocalories per mole only. So valinomycin competes less effectively with water for sodium ion. Valinomycin as a carrier ionophore has a good turnover and transports potassium ion many times per second, returning each time to surface to pick up a new potassium ion. High flexibility of valinomycin makes this a facile process. Gramicidine A is a 15 amino acid residue long peptide with alternate L and D amino acids. All amino acids are hydrophobic with both N and C terminals being blocked. X-ray diffraction studies have revealed that the gramicidine A exists as a dimer with head-to-tail joining. The result is a 4-angstrom diameter helical channel for sodium and potassium ion movement. The hydrophobic amino acids allow formation of the nonpolar exterior in contact with membrane structures. Ions enter and diffuse to the other side. In this mechanism, the channel former does not move. The two kinds of ionophores, carriers and channel formers, can be distinguished experimentally. This is done by measuring ionic conductance of a synthetic bilayer of lipids as models. The ionic conductance is measured as a function of temperature. The temperature range is around the transition temperature of the lipid, at which lipid bilayer becomes highly fluid. In case of channel formers, membrane fluidity does not matter. On the other hand, a carrier ionophore has to move through the membrane. Its movement is affected by the membrane fluidity. The data shown here was obtained with valinomycin as a carrier and gramicidine as channel former. Conductance measurements have shown that a channel is open for about 1 second. The opening and closure are spontaneous processes. The channel is selective towards monovalent cations and does not allow either divalent cations or even other monovalent anions. More than 10 to the power 7 ions move across a channel per second. That is only 10 times less than free diffusion in the water. On the other hand, the carriers transport ions less than 10 raised to the power 3 ions per second. Spectroscopic and X-ray diffraction studies indicate that the dimer is in equilibrium with the monomer. The pore formed only by the dimer is lined with carbonyl groups of the channel former. The alternate L and D configurations are necessary in this design to place the hydrophobic side chains outside. The coordination of the moving cation by the carbonyl groups is transient at each step of the movement. The ions and many small molecules pass from one cell to another in prokaryotes and eukaryotes via large aqueous channels which are part of the gap junctions. Revel and Karnowski discovered gap junctions as discrete clusters in plasma membranes of the connected cells. The central lumen, the hole, of gap junctions is of 20 angstrom diameter. The density of the channels in the gap junction is 28,000 per micrometer square. The centers of the neighboring channels are about 85 angstrom apart. It was Werner Lowenstein who in 1964 used the fluorescent molecules to find that polar molecules of less than 1 kilodalton can pass through channels of the gap junctions. Such channels are formed by 12 molecules of a 32 kilodalton protein connexin. A cell-to-cell channel remains open from seconds to minutes. Calcium ions, protons, and many hormones regulate the opening and closure of the channel in a cooperative process. The cells also need to transport larger molecules. These transports follow very different mechanisms. One such mechanism is exocytosis. Many proteins, hormones, and neurotransmitters have to move out of the cell. These pass through cell membranes into the extracellular space by exocytosis. In exocytosis, such molecules fuse with the plasma membranes in the constitutive secretory pathway. Golgi releases vesicles which include these molecules and fuse with the membranes. All cells have this pathway. In cells which are involved in this rapid release of molecules, in response to stimulus such as insulin or digestive enzymes by pancreatic cells, neurotransmitters by nerve cells. In such cases, these molecules are also stored in secretory vesicles by budding from trans-Golgi network. Coated vesicles are so called as these have specific proteins on their cytosolic surfaces. Clethrins coat vesicles involved in transport of molecules from Golgi to plasma membrane and from plasma membrane into the cell. COP1 and COP2 are also involved in transport within Golgi compartments and from Golgi to rough endoplasmic reticulum. Endocytosis is the process in which macromolecules from outside the cells are transported into the cells. The material is first enclosed by local portion of the membrane. Invagination and pinching off to form intracellular vesicles follows. Phagocytosis is a subclass of endocytosis in which even larger entities such as bacteria or cell debris enter cells as bigger vesicles called phagosomes. Here the initial step of engulfing usually requires a specific cell receptor, while blood cells such as macrophages, neutrophils use phagocytosis for cellular protection. Phagosomes fuse with lysosomes to be destroyed. Small, uncharged or hydrophobic moieties, water, carbon dioxide, oxygen, many other gases, urea, ethanol, and steroids. Such moieties can cross the membranes by simple diffusion. This does not normally require involvement of any integral protein as a transporter or carrier, nor it involves any specificity. In such cases, diffusion rates are driven by concentration gradient of that particular molecule across the membrane. Unlike facilitated diffusion, which is also passive but does require involvement of a specific integral protein as a carrier or transporter, the rate of simple diffusion versus concentration gradient does not reach saturation. Water has been mentioned as a molecule which can travel across membranes by simple diffusion. That is true in case of most of the cell membranes. Many cells, however, require rapid transport of hydrogen molecules across their membranes. Examples of such cells are erythrocytes and cells of the kidney. Water is a neutral molecule of small size and is normally present in high concentrations in most of the cells. In these special cells, water movement is crucial and needs a channel protein. Water channel proteins are called aquaporins, which help water with facilitated diffusion and accelerate its transport. Aquaporins are tetramers of 28 kilodalton subunits. Each subunit has 6 transmembrane helices. Each subunit has a central pore. It is estimated that a large number of deaths, especially those of infants, occur because of dehydration. Diarrhea due to cholera is the most frequent cause. A simple oral rehydration therapy can prevent these deaths. In its simplest form, a solution of glucose and sodium chloride is a good rehydration agent. Simple water is not effective as the large intestine is secreting both water and sodium ion. Given glucose plus sodium ion, glucose facilitates sodium uptake through the sodium ion glucose symporter. which enhances water movement through the epithelial cells of the intestine into the blood. This importer transports glucose or even other sugars or amino acids from to concentration in intestinal lumen to high concentration in the cytosol of the epithelial cells. In this ion-driven active transport, the energy is derived from the transport of the sodium ion down the concentration gradient. The glucose concentration gradient is maintained by facilitated diffusion of the glucose to outside of the cell by a uniporter called glucose transporter. The low concentration of sodium ion inside the cell is ensured by sodium ion potassium ion ATPase, which as discussed before is an ATP driven active transport. Thus oral rehydration therapy works because of so many transport mechanisms in place. Cystic fibrosis is actually caused by a defective ion channel. Cystic fibrosis can cause obstruction of both gastrointestinal tract and respiratory tracts. It often leads to bacterial infections of the airways and death by the age of about, or even before, by the time one reaches an age of 30 years. It's a hereditary disease and serious symptoms ensue if both copies of the genes are defective. Very thick mucus is present inside lungs, and this breeds bacteria like Staphylococcus aureus and Pseudomonas aeruginosa. The gene responsible codes for a membrane protein called cystic fibrosis transmembrane regulator, abbreviated as CFTR. It has a 12 transmembrane helices and is structurally analogous to the multi-drug transporter discussed earlier. The extracellular part is glycosylated with oligosaccharide chains. The cytoplasmic component has three distinct domains, NBF1 and NBF2, which are nucleotide binding folds and which bind to ATP. The third is the regulatory domain, which can get phosphorylated by a cyclic AMP dependent protein kinase. The most common mutation which causes cystic fibrosis is the deletion of phenylalanine at 508 position in the NBF1. The normal CFTR is an ion channel for chloride anion. The phosphorylation of the protein facilitates the channeling of chloride anion. The commonest mutation leads to misfolded CFTR1, which does not get inserted in the membrane. In other defective genes, the phosphorylation is prevented. In all such defects, the channel is non-functional. In the epithelial cells lining airways, the digestive tracts, and even exocrine glands like pancreas, sweat glands, bile ducts, and vas deferens, the non-functional channel leads to serious consequences. Epithelial cells present on the inner surface of the lungs secrete a substance which traps and kills bacteria. This is part of the innate immunity in humans. The cilia on these cells go on clearing the surface by sweeping away the resulting mass. In cystic fibrosis patients, the non-functional chloride channel cannot transport chloride anion out of the cells. So the sodium chloride concentration outside these epithelial cells is low. The substance which kills bacteria apparently requires high sodium chloride concentration and hence fails to exert bactericidal action where sodium chloride concentration is low due to defective channel. Hence, recruiting infections in individuals with the defective channels by Staphylococcus aureus and Pseudomonas aeruginosa damage the lungs. Their respiratory efficiency decreases. CFTR1 is an example of ABC transport. Such transporters have ATP binding cassettes. In fact, ABC transporters are a family of multi-drug transporters as they actually transport many ions, amino acids, vitamins, and bile salts across plasma membrane. In this module, we have learnt about the following concepts and processes. We learnt about glucose transporters, bicarbonate chloride anion exchange protein. We learnt about ion channels, and finally we learnt about cystic fibrosis, its molecular basis. While small lipophilic molecules can easily pass through membranes, hydrophilic or charged molecules require mediated transport. Passive transport by facilitated diffusion, primary active transport, which is ATP driven, and secondary active transport, which is ion driven, are various modes to achieve that. The discussion on oral rehydration therapy and cystic fibrosis illustrated how important role membrane transport plays in diverse biological processes. Thank you.